In this video, we are going to talk about the different allosteric effectors, inhibitors, activators that go along with the second half of the metabolic pathways. So mostly the Krebs cycle, but we're also going to go over a quick little activator and inhibitor of lactate dehydrogenase, which is a part of the Cori cycle. So let's jump right in. Let's start off with lactate dehydrogenase. So there are two different types of this enzyme. Lactate dehydrogenase 1, or also called H4, is found in the heart. For this type of the enzyme, lactate activates it, and it prefers aerobic mechanisms. And it also has a higher ATP yield. Now, the second kind of this enzyme is lactate dehydrogenase 2, or also known as M4. And this is in skeletal muscle. So for this one, pyruvate activates it. It favors anaerobic metabolism. It speeds up glycolysis, and this occurs during exercise. Okay, so that is all it is for the different types of lactate dehydrogenase. So now we're going to move on to the transition step and then the Krebs cycle. So for the transition step, there are two enzymes called PDH kinase and phosphoprotein phosphatase. And these two enzymes phosphorylate and unphosphorylate the pyruvate dehydrogenase complex. So I'm going to write down these two. PDH kinase, this phosphorylates. And this makes an inactive form. of the pyruvate dehydrogenase complex. Now, the second enzyme is phosphoprotein phosphatase. And this dephosphorylates, and it makes an active form of the dehydrogenase complex. So those are just two different enzymes that have covalent control over this complex. But now if we're moving on to allosteric control of this complex, I'll put an arrow down here. It's inhibited by ATP, NADH, and acetyl-CoA. And this makes sense because these are all products of either the transition step or the citric acid cycle. So if there's a lot of the products, we're not going to want to activate the enzymes to create more of those products. Now, this complex is activated by ADP. So if there's a lot of ADP, that means there's not a lot of ATP, which means we need to make more ATP. And we're going to do that by going through the Krebs cycle and going through that transition step. So it's going to activate this complex to hopefully start the Krebs cycle. Okay, moving on to the Krebs cycle itself, there's really only three enzymes that we're going to focus on. First is going to be citrate synthase. And for this one, it's just inhibited by ATP, NADH, succinyl-CoA, and citrate. Now, this should also make sense because these are all further down products of this pathway. So if there's a lot of them, we're not going to want to make more. Moving on, the next enzyme that we're going to focus on is isocitrate dehydrogenase. Now, for this one, inhibitors include ATP and NADH, while activators include ADP and NAD+. So these are just the opposite. Again, ATP and NADH, they're both products, so they're most likely going to inhibit this enzyme. And then the opposite, if there's a lot of reactants, it's going to activate it so I can go ahead and process those. Now, the last enzyme that we're going to focus on is A-ketoglutarate dehydrogenase. We'll go ahead and list out the inhibitors, ATP, NADH, and succinyl-CoA. And it's activated by ADP and NAD+. Very similar to the one above. So those are really all of the allosteric effectors of the second part of metabolism. Remember, these two enzymes here for the transition step are covalent control because they will be adding a phosphate and taking off a phosphate. But other than that, the most important thing to remember is that most likely a high amount of products will inhibit that enzyme. And also for lactate dehydrogenase, there are those two different forms, one for the heart and then one for the skeletal muscle. Different things activated, different things inactivated, kind of deciding which way on the pathway it wants to go, because if we remember, lactate dehydrogenase can take pyruvate to lactate and also lactate back to pyruvate. So those are all of the allosteric effectors for our biochemical metabolism pathways. In the next video, I'm going to go over the pentose phosphate pathway really quickly, and then kind of just an overall overview of everything we talked about. Again, the notes for this video will be in the link tree, and that is all I have for you today.